Today we are going to talk about CHF, congestive heart failure. CHF. So in this we are going to talk about what is CHF, the drugs and drugs mechanism, what is the mechanism of drugs, and in this what types of drugs we are going to talk about is the cardioglycosides and its structure, uses, and also one more thing, SAR, structure activity relationship. Then we will talk about other drug like nesretide, which is a hormone, natriuretic peptide hormones. Then we're gonna talk about third drug, ET antagonist, endothelian antagonist, receptor antagonist, in which the drug is bosentan and tazobactam. We have a more, but these are like most commonly used. So now we will talk about what is CHF. CHF, drug used in CHF, congestive heart failure, is when the heart is not able not able to pump the blood out of heart. Okay, not pump, which is not able to pump out the blood, like the concern, the contraction is not enough. So now we'll talk about here CHF. We have three types of CHF. First is left heart failure, second is right heart failure, and third is both side heart failure. Most commonly is left side, most commonly, in which the left part is not able to sufficiently work. It's not able to sufficiently work properly, so it will cause problems. Common most only left part of heart. So in this, like, the blood is gonna see, this is left and right part. In this like a left is not going to pump out, so blood is going to be inside, not able to, so blood is going to be inside the heart. So if blood is not pumping outside the heart, so it will not go into the other body parts. Blood will not go to other body parts, which will cause like problem like oxygen supply, nutrient supply, and other problem. Then we have a right failure, like in which the right part of the heart is not able to pump or contract, like not able to contract. Okay, then we have a both side, like both part of the heart is not working properly. And also, like, one more thing, the normal heart rate is 120 by 80 mhg, and this part is systolic. Systolic means contraction, and this part is diastolic. It's called diastolic, and this is diastolic, in which heart relax. So like the common thing you can see here, or like the mnemonic I would say, when you like contract, it need more power. So like the value is upside is high, then below side denominator. So it's contract, systole, contract. That means blood goes out from heart. Okay, okay, now we will gonna talk about the drugs Mainly we are going to talk about two types of drugs. Two types of drugs in cardiac glycosides. First is digoxin and digitoxin. They both are cardiac glycosides. Means cardiac means like heart, and glycoside is the secondary metabolite obtained from plants which contains which contains sugar moiety and non-sugar moiety. Which is non-sugar moiety is called aglycon, and the sugar moiety is called only glycon. Okay, and they both are obtained from Digitalis purpura, which is a foxglove plant, also called foxglove plant. Now we are talking about structures, digoxin and digitoxin. They both have similar structure. The only difference is that in digitoxin has OH group at carbon 12 and digitoxin lacks OH group at 12 position. The only difference it is has. Now I'm going to tell you the trick. See, trick or like method, you can say. How could you go? First we will divide the structure as it contains two parts: sugar moiety and non-sugar. Okay, sugar part which it contains is called, in case of digoxin, sugar part is called digitoxis, beta d digitoxis. Digi is same, digoxin and toxis. Okay, and it is a three molecules of beta d digitoxin, and non-sugar moiety is deoxygenesis. Deoxygenin. Now in the digitoxin, sugar, non-sugar, sugar part is same in both three molecules. This 3 into beta d digitoxos. You can remember like the sugar part is same and the non-sugar part is different in both. In this it is digitoxy, then genin. The whole thing is same. Just remove the N part. And add genin. Okay, now I will tell you the structure. We're gonna make only one structure in which both contain the same part. So first, sugar and non-sugar moiety. So first we can make the important part, which is non-sugar moiety. Non-sugar moiety is mainly has main effect in like lipophilicity, half-life. Like it's the active part of active part of the compound, and it shows like pharmacokinetics of the drugs. You could say that. I will do here. Non-sugar part is involved in pharmacokinetics. And sugar part is involved in pharmacodynamics. Like kinetics, like ADME, absorption, digestion, metabolism, and excretion. Like the kinetic means movement, so means the compound movement, how it moves when we take the medicine or take the compound in our body, and how it is metabolized, absorbed, distributed, then excrete out eventually. So non-sugar part is important. So in this, if we are going to talk about the main thing we'll be talking is non-sugar. Even if you omit the part of non-sugar sugar part, then it's not important. So non-sugar part is very important when we talk about the SAR of the drug or mechanism of the drug. Main thing, the thing which is going to affect is non-sugar part of the drug. Now structure. It have like 5 ring structures, and in this one we have a lectone part in the, okay. So I will show. First make one cyclohexane, then other cyclohexane which is like attached. Then we are going to make little bit upside. This is like anthracene but without double bonds. Now you have three cyclohexane. Then what are you gonna do? Now you're going to make adjacently 5 ring compound. Now you have a 5 ring, then you will make again one 5 ring compound. 5 ring compound again, but in this you have to make oxygen. This is not a complete 5 ring, this is a heterocyclic 5 ring, okay. And the position of this oxygen is 1. You cannot say exactly 1. Like it have one more group here. No, not here. It has one double bond in this and this. So this is up. Now we can count 1, 2, 3, 4, 5. 5. So oxygen should be at 4th in the heterocyclic ring, and this is, and at 3rd it should have a ketone group, and at 1st position it should have double bond. And this part is, if you can recall or remember the structure, this part is lactone. Like lactone is like this, then you have this, then this part is lactone, and this we have a phenyl ring, we have a ring or whatever you want to attach to it. So we have A, B, C, D, E. Now I will like add the extra parts so you can remember. This is important. After this we have a sugar moiety. Sugar is attached. Okay, so this is very important in SAR. I will tell you when I'm going to talk about SAR. So now you have to make CH3 group. This line means CH3. I'm not gonna draw write CH3 every time because it will make the structure hard. CH3 between the rings. If you can see, this is like between the rings. So now we will again gonna add between the rings CH3. Okay, now we have completed with CH3. The both side CH3 is on upside. Now we will add OH, 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 OH. This is OH, and now opposite OH, OH and OH. You will understand later when I'm going to talk about the SAR that the structure is cis, trans, cis. Like A and B is cis to each other, and B and C is trans, B and C and D is cis to each other. So you will know how the structure is opposite, and if we can have different structures, different like stereochemistry, but we are not gonna talk about and make things more difficult in our case. So I will just gonna stick with this. So this whole parties are non-sugar moiety, which is, if we are talking about digoxin, the non-sugar moiety will be digoxygenin. Okay, now I will do counting 1, 2, 3, 4, 5, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. No, I don't remember the. Wait, let me see. 11, 12, 13, 14, 15, 16, and 17. Or 17. Now I will tell you this part. This is 12th position. If we remove this hydroxyl, it will become digitoxin. If we add this part, if we add this hydroxyl group, it will digoxin. Okay, now we will make sugar part in case you might need the in future. If you want to add draw, we can draw. So let's add. This is very easy. This you have already known. This is the 3 molecule parts of digitoxin. So means the 3 molecule of the same structure. So just we need to know how we are gonna add this part. First, draw a heterocyclic cyclohexene, and this is the overall structure. Like this is the basic. If I will draw here for reference, so you can identify. This is H3. This we will attach from this part and this part. Attachment. Okay, now we will add again on this part. This is little bit tricky, but eventually we'll get hang of it. So you can understand. I will give just little bit. This is O, CH3, O and CH3. You might be thinking, oh, this is different. This doesn't seem like, but no, it's all structures same. Like we are just like rotating each part sometimes. See, as what we are doing, first the O starts from upside, then it go downside. So we need to turn the structure also. So this is our basic structure. This is our sugar moiety. Sugar, 3 sugars of digitoxos. Now, since we have formed the structure, we can also talk about the SAR. SAR is structure activity relationship. Now I'll show the structure. SAR, structure activity relationship. What is the relationship of a drug structure in pharmacodynamics, kinetics? So we have in cardioglycoside we have 3 structures, but mainly we're going to talk about digitoxin and digoxin. See, digoxinogenin, presence of OH at C12, this is sensory, and detoxygenin, presence of OH at 660. This is not important. Main compound we usually talk about digoxin and digitoxin. They are very important structures. So now this is our glycon, aglycon. This is our main structure. So we have around 9 SAR, which is very important. First, most important is replacement of OH at C14. Where is C14? This. If we remove the OH from C14, it will inactive the compound. It will lead to the inactivity of compound. If decreasing the activity is fine, but if you have no OH at 14th position of a glycon moiety, then it will inactive compound. Then it will make the compound inactive, which is no use. Second is that's the most important. If you're not gonna write this part in exam, you might lose. Then we will talk about third OH. Beta OH is involved in glycosidic linkage. 14th B, very important. Now we'll talk about C, L, A and B. Ring junction is cis. I already told you, cis, trans, then cis. Cis AB cis configuration is not mandatory. Not mandatory. B and C cis trans. You can talk about cis transes. Cis transes ring junction of cis trans makes the nucleus very stable and active. The presence of lactone ring at C17. The whole structure is lactone. I already told you. C1, 2, 3, 4, 5. Lactone at C17. Here is C17 marked. Is very important. Aglycon or sugar moiety increase the. What is the role of then sugar? If you are gonna just talking about glycon the whole video, the importance of the glycogen is absorption, solubility, distribution. Very important, we must say. Then the presence of additional OH in any part of glycon have a significant impact on the partitioning and pharmacokinetics for each glycoside. If we have a more oxygen OH group, hydroxyl group at any different position of our glycoside, it will increase our pharmacokinetics. Like this, in this getoxygenin, we have a presence of more one oxygen hydroxyl group at C16. Like this part, we have one more hydroxyl group, so it is increasing its activity and making more good partitioning and pharmacokinetics ability. Now we are not complete with it yet. Now we will talk about The SAR is complete. Now we'll talk about mechanism of action, which is the most awaited part of this video. Mechanism action of only cardioglycoside. Cardioglycoside. Very basic. Inhibits sodium, potassium, ATPase enzyme. We are going to talk about the mechanism now of cardioglycosides. Cardioglycosides inhibits sodium potassium ATPase enzyme. It means sodium in exchange of potassium will use up the energy. ATPase means ATP, which will use the energy. Then it will going to exchange or transport the sodium and potassium. In normal cases, first potassium will come inside of the cell, then sodium will go outside. Simultaneously it will occur, and when sodium is outside, means the sodium will has less inside as it is going outside. So by exchange of sodium by sodium calcium exchanger, sodium will come inside the cell and calcium will go outside the cell. Okay, in normal cases that's how it's occur. So the digitalis will come and blocks the sodium ATPase pump, sodium potassium pump, ATPase pump. So there will be no influx of potassium inside the cell, means there will be more potassium outside the cell, and then no exchange of sodium outside. No outflux of sodium means there will be lots of sodium inside the cell, means there is more nice sodium, means now we have to get out the extra sodium outside the body, outside the cell. So what we are gonna do, we are gonna transport sodium by this sodium calcium exchanger. And how we are going to do that? For we are gonna reverse this. Like initially the sodium is coming inside, but now we will gonna change it to sodium outflux, means sodium will going outside, not inside. So to make it equal, the calcium will not go outside, it will come inside. Calcium will gonna come inside the cell, means now the sodium is going out, the sodium level is going to be maintained, and the calcium ions are coming inside the cells, means the calcium is increased concentration of. And as we know, calcium is used in contraction of muscles. So our main agenda was to increase the contractility of heart. If we can increase contractility, means it will gonna pump more blood outside heart. So the blood is pumping. Now the pumping of blood is sufficient for our overall body. Now the calcium will come and bind on SR and cause this is myosin, this is muscles, myosin and filaments, and those whole calcium will go inside and cause more contraction of this. That's how it works. Calcium controls the force of contraction. So we are indirectly increasing the concentration. So make sure to know that we want to just increase the calcium concentration in our cell, and we are doing it by first stopping the sodium potassium ATPase, increasing sodium in our cell, in our cell. Then the exchanger will gonna work opposite as they work usually. Sodium is going outside, calcium is inside. It is increasing, calcium is going to SR. Then it will cause the contraction of heart muscles and it will works. Now we are done with mechanism. Now the extra part we are gonna talk about is neseritide. Is a natriuretic hormone. It is often found human. It is made from recombinant DNA, rDNA, recombinant. Now we have an endothelium antagonist, receptor antagonist. Bosentan has a more affinity to ETA receptor. Receptor means it will block more this. And tazontan is non-selective. Non-selective means it will blocks both ETA and blocks ETA and ETB. Structure is nice, quite good. There are prefix ET है receptor, ET receptor, suffix you could remember by san. Then the structure is quite similar of both the drugs. First I will make bosentan and I will tell you simultaneously uses, so there will be no problem. First we are gonna make structure. It is a sulfonide group. We have sulfonide group. First we will make benzene ring, then sulfonide attached with NH. Then on the fourth position we will gonna make plus. This is not plus but isobutyl, but we could say plus. Then little bit complex structure we have. This is little bit hard to remember, I must say, but it will be fun to draw if you are able to draw in exam. I must say this is really fun part of this structure. Now I will make tazontan. Same structure, benzene, sulfonide, sulfonide. Then we have a same nitrogen complex structure. Now we have oxygen, benzene, OCH3, then CH2. Oh, this part is same. This part is same. The only part is different is 1, 2, 3. This is little bit difficult. Just this part is different and this part is different. So what are the different parts? This is different part, this is different part, or else is everything same. And the one thing is here we have not benzene but N structure. The only we have 3 difference in this. See, this is isopropyl, this is isobutyl. Only we have and this is little bit more complex structure, and everything else is same in this presence of not a benzene but a heterocyclic ring. Now I will tell you the uses. Uses are nice, I must say. You should remember this. Little different. Uses are used in boson 10, used in management of. It does not treat but manage pulmonary hypertension, which is nice, I must say. And second is treat systemic scleroderma, sclerosis or scleroderma, or systemic sclerosis. Now we have a use of tezos, and then vasodilator. Both are vasodilator. Used in acute heart failure. Mechanism of endothelin receptor antagonist are drugs binds to ETA or ETB hormones receptors. ETA or ETB receptors, and it binds, blocks, and prevents constriction of the vessels. Endothelin receptors are present on vascular smooth muscles. So in normally it's called endothelin receptor prevents vasodilation and cause constriction. But if we block the endothelin receptor, so and now it will cause vasodilation, and vasodilation is used in our congestive heart failure to treat. And thank you guys for our overall video. This is our overall. For any queries, make sure to message.